Hello, and welcome to the BioConference Live webinar on heavy light chain combination assays, what laboratories should know about M protein detection. Our speaker today is Dr. Richard O'Hara. Dr. O'Hara received a baccalaureate degree in biology from the University of Texas at Austin. After working as a research associate for a few years, he entered the immunology graduate program at the University of Texas Southwestern Medical School. Dr. O'Hara spent the final 2 years of his graduate training at Dalhousie University in Halifax, Nova Scotia, working with the group that established the first liver transplant program in the region. Following his graduate training, he entered a postdoctoral training program at Harvard Medical School, working on mechanisms of T-cell mediated immune regulation. He entered the pharmaceutical industry, joining Genetics Institute as a research scientist in 1988. His work on cytokine biology led to the development of 2 new therapeutic drugs and several U.S. patents in hematology and oncology. Later work in the role of co-stimulation in graft rejection and autoimmunity led to the development of several other drug candidates. In 2007, after more than 20 years of research in the fields of autoimmunity and transplantation, he joined the Binding Site as Senior Director of Scientific Affairs. In his role at Binding Site, Dr. O'Hara has presented extensively at major U.S. academic medical centers on the characterization of monoclonal proteins in patients with multiple myeloma and other plasma cell dyscrasias. Before I turn it over to Dr. O'Hara, I want to cover a few logistics. During the presentation, you have the ability to type questions into the Q&A box located in the lower left hand of your screen. After his presentation, Dr. O'Hara will have time to answer the questions. Dr. O'Hara, I will now turn it over to you.
Thank you, Maureen, for that introduction, and thanks to everybody for joining the broadcast today. What I wanted to talk about were some new tools in the toolkit that the laboratory can use when detecting monoclonal proteins from diseases such as multiple myeloma and other plasma cell dyscrasias. Before we jump into the heavy light chain combination assay, let's just take a couple of minutes to review immunoglobulin structure. Most of you are certainly aware that immunoglobulins are a heterodimer made up of 2 heavy chains and 2 light chains. The antigen binding site is encoded by the variable region of both the heavy and light chains. We're not going to spend much time talking about antigen binding today. There are 5 heavy chain combinations, 5 heavy chain isotypes rather, IgG, IgA, M, D, and E, and 2 light chain isotypes, kappa and lambda, giving us a possible 10 heavy light chain combinations, such as IgG kappa, IgG lambda, etc. Now, I want to spend a couple of minutes talking about the traditional tools for detecting monoclonal proteins in plasma cell dyscrasias. On the left-hand side, you see a serum protein electrophoresis gel from a patient with multiple myeloma having a prominent end spike. Looking at the gel on the lower left, you can see a distinct band constituting albumin, and then moving from left to right, alpha 1, alpha 2, beta, and gamma regions. Note that in this particular SPEP gel, a distinct and dense band is formed in the gamma region instead of the normal heterogeneous smear you would expect from diverse immunoglobulin. This is because of monoclonal protein being produced and gives us the resulting M spike in the gamma region that you see on the densitometry trace up above the gel. If this patient were, for instance, an IgG lambda multiple myeloma patient, the immunofixation might look something like you see on the right-hand side. Again, from the left side of the gel, you see a total serum protein band with a very distinct band in the gamma region. Following over, stains for IgG and lambda denote this patient as an IgG lambda monoclonal protein producer. About 10 years ago, the FDA approved the Freelite chain assay, which detects immunoglobulin light chains that are not bound in intact immunoglobulin. As I mentioned, it was FDA approved in 2001 under the trade name Freelite. The Freelite chain assay detects all light chain only myeloma patients at diagnosis, in contrast to the 50% of light chain only myeloma patients that are detectable by SBE. It also detects approximately 90% of intact immunoglobulin myeloma patients at diagnosis, owing to the fact that they produce excess free light chains, as do most plasma cells. When it comes to monitoring patients, the short half-life of the analyte measured by the Freelite assay allows a real-time assessment of tumor kill during therapy. We'll discuss a little bit more about free light chains and their short half-life as we go through the talk. The Freelite assay was incorporated into the International Myeloma Working Group guidelines in 2006 and was also incorporated into the National Comprehensive Cancer Network guidelines in 2010. Now, these three assays, when combined together, provide a very powerful tool for the laboratory and the pathologist in terms of detecting monoclonal proteins. But like all serum assays, they have certain limitations. For serum protein electrophoresis, we know that not all monoclonal bands migrate in the gamma region. We know that this can make them difficult to quantitate at low levels or when the bands migrate unusually. For immunofixation electrophoresis, bands may be easy to discern, as I showed you in the previous gel, but they don't all look like that. Because of this, sometimes it's hard to call the presence of a band, especially when proteins are present at low levels. Even if the proteins are readily distinct, we know that immunofixation is not a quantitative assay. It can tell the pathologist that the patient is making monoclonal protein and what the isotype of that protein is, but it can't tell anybody how much, and therefore it can't give us any information about tumor load during therapy. The Freelite assay, while detecting all light chain myeloma patients at diagnosis and detecting over 90% of intact Ig myeloma patients, does in fact not detect changes in about 10% of intact immunoglobulin patients because they don't make excess light chains. So what I wanted to talk about specifically today was the heavy chain light chain combination assay known as HeavyLight. HeavyLight works on the premise that we know going in that the free light chain assay provides sensitive detection of tumor-derived M protein. We know that the ratio between the involved and the uninvolved light chains provides evidence of clonality in a patient with monoclonal gammopathy. For instance, in a patient with a kappa myeloma, we know whether it's an an intact kappa or a free kappa myeloma, we know that not only the concentration of the kappa protein is important, but the uninvolved lambda chain also adds meaning when it comes to determining clonality. The HeavyLight assay sought to answer the question, could an assay which detected individual immunoglobulin heavy chain light chain pairs provide similar advantages when measuring intact M protein? So we're actually very excited about the prospects for HeavyLight. The assay was just FDA approved this past December. And what I wanted to talk about was what these antibodies specifically are. HeavyLight recognizes conformational epitopes between heavy chain and light chains. When those two chains combine to form an intact immunoglobulin, their physical space makes them present conformational determinants which can be recognized specifically by HeavyLight antibodies. These antibodies recognize epitopes that are unique to each heavy light chain pair. Thus, they can distinguish IgA kappa from IgA lambda, IgG kappa from IgG lambda, and IgM kappa from IgM lambda. Assays which will distinguish IgD and IgE are still in development. One of the questions that comes up is, well, then HeavyLight is simply a replacement for Freelite. And I'll show you some data to indicate later that that's definitely not the case, because these antibodies recognize different things. As I mentioned, HeavyLight recognizes epitopes that are formed when heavy chains and light chains pair. Freelite, on the other hand, recognizes immunoglobulin light chains that are not bound in intact immunoglobulin. The epitopes recognized by the Freelite reagent are not available for binding when bound to heavy chain, and thus only recognize free circulating light chains. Now, if you're going to make an assay that measures individual immunoglobulin heavy chain light chain pairs, then you need to rest assured that when you sum up the individual pairs for any given heavy chain isotype, that those will equal the results you get when you measure total IgG, for instance. So in this slide, what you see is IgG and IgA on the top half of the slide and total IgM on the bottom half of the slide. All of those are represented on the x-axis. On the y-axis for each graph, you see the sum of any individual heavy chain light chain pair. For instance, in the upper left, you see the sum of IgG lambda and IgG kappa represented on the y-axis measured against total IgG. The lines of identity for each heavy chain isotype running through the graphs let us know that when we're measuring by HeavyLight IgG kappa plus IgG lambda, for instance, we are in fact capturing all of the IgG. Normal ranges have been established for HeavyLight reagents, and I'm not going to go through each one of these individually as they are present in the package inserts, but I do want to point out that like Freelite, one of the tools that we make use of when using HeavyLight is the ratio between individual heavy chain light chain pairs. Down the right-hand side of the table, you can see the normal ratios for each isotype. The ratio for G kappa over G lambda runs from approximately 1 to 3, as it does for IgM kappa over lambda. For IgA kappa over lambda, the ratio for the normal range is from slightly under 1 to just under 2. Now, I want to point out, as it will become evident later when we look at a case study, that changes in the heavy light chain ratio are the result of changes in either the concentration of the involved heavy chain light chain pair or the uninvolved heavy chain light chain pair. So, for instance, in a patient with an A kappa myeloma, you might see an increase in the A kappa over A lambda ratio because the A lambda was decreased. And I'll show you an example of that as we move forward. One of the things that I want to emphasize as we go through this today is that unlike SPE and IFE, both Freelite and HeavyLight read within a normal range. That is to say that everybody listening to the broadcast today would have a free kappa and a free lambda value that can be read, hopefully within the normal range. And the same is true for HeavyLight for IgG, A, and M. Because we can read normal, this means that we can sensitively read abnormal. And that becomes important when we want to talk about patients who might be being treated and are entering into remission. We want to measure all the way down into the normal range. Similarly, it can tell us about residual disease, and it can also tell us about early relapse. So let's talk for a minute about factors which can affect monoclonal protein measurement. One of these, especially when it comes to measuring IgG, is the variable half-life of immunoglobulins. As you can see from this table, looking at IgA and IgM, you can see that they have a half-life which is relatively similar to most normal serum proteins of around 6 to 8 days. Interestingly, free kappa and free lambda provide a monitoring advantage when measuring free light chains because the half-life of the analyte, either the free kappa or the free lambda light chain, has a very short half-life of only 2 to 6 hours. However, IgG has a half-life of almost 3 weeks. And this can compound measuring, especially when we're talking about IgG myelomas. The reason for this extended half-life of IgG has to do with a naturally occurring receptor known as FcRn. Some of you may know about FcRn. It's the receptor, it's FcRn standing for Fc receptor on neonatal tissue or neonatal placental interface tissue. This receptor specifically binds IgG and is the receptor which is responsible for transferring IgG across the maternal fetal interface at the placenta, thus conferring passive immunity to the fetus during gestation. Well, all proteins are taken up by endothelial cells to be degraded and into peptides and amino acids, and this is true too for circulating IgG. However, once it's in the endosome, or actually on the surface of the endothelial cell, IgG binds the FcRn receptor, and that changes its intracellular trafficking. Instead of being combined with a lysosome and degraded proteolytically, FcRn-bound IgG is protected from proteolytic degradation and is trafficked back to the cell surface where it's re-released into the circulation. The result of this is an extended half-life for IgG due to the fact that it's protected from proteolytic cleavage. This process, however, is concentration dependent. At normal concentrations of IgG, around 10 to 15 grams per liter, the FcRn receptor is fully saturated. This ends up giving us a half-life for IgG of approximately 21 days, as you can see on this graph. However, in this data, which was generated, as you can see, quite some time ago by Herman Waldman and Sam Strober, using radiolabeled IgG injected into patients, a study that we can't possibly imagine doing today, you can see that at high concentrations of IgG, up around, say, 60 grams per liter, that saturation of the FcRn receptor has made a lot of this protein no longer protected from proteolytic degradation. Therefore, the half-life drops down to about 10 days. Similarly, if you look at the other end of the curve, you can see that at very low concentrations of IgG, that the half-life can be extended out dramatically out to 60 or 70 days. So at about 2 grams per liter, for instance, for IgG, that half-life is extended dramatically because there's plenty of FcRn receptor available for binding. Now, this actually gives us some insight into two questions that frequently show up during laboratory detection of monoclonal proteins. The first is, okay, let's see if I can get, the first is, sorry, a little technical problem, has been asked on occasion, well, if the half-life of IgG is 3 weeks, then how does my patient with 60 grams per liter of monoclonal protein drop down to 10 grams per liter after a single therapy cycle? If a therapy cycle is 3 weeks to a month, and you have a protein with a half-life of 21 days, it shouldn't get that dramatic a drop. But as you see from this graph, at 60 grams per liter of monoclonal protein, the half-life is down to 10 days. So until it drops back down into more physiologic concentrations, the half-life of the protein is much shorter, and you can get much more dramatic drops. At the other end of the scale comes the answer to the question of, if Freelite is more sensitive than IFE, which it is, then why does IFE remain positive when the Freelite chain test becomes negative? Well, again, you can imagine for a patient with a monoclonal IgG protein that would move as a single electrophoretic entity on an IFE gel, that a low level of that protein would be relatively easily detected on an IFE gel. Because this protein is present at low concentrations, it's continually protected by the FcRn receptor, and yet it shows up as a band because it is a single electrophoretic mobility. Another thing that we know happens in multiple myeloma patients, for instance, that affects their measurement of monoclonal protein is that there are changes in plasma volume and hematocrit. Patients with compromised renal function can have dramatic increase in plasma volume. Patients who have disease exacerbations may have anemia resulting from plasma cell occupying that space in the bone marrow. Well, if you've got a test such as SPE or total immunoglobulin that are concentration-dependent measurements, you can see how an increase in plasma volume or a decrease in hematocrit could change the relative concentration of such a protein. But if you're dealing with a ratio between a monoclonal protein and its uninvolved isotype partner, you could see, as represented on this slide, that if you, for instance, had a concentration or a ratio of IgG kappa to IgG lambda of 3 to 1, that concentration, that ratio, would remain independent of both plasma volume and hematocrit. So just to sum up, monoclonal protein measurements can be affected by catabolism of the IgG, of the immunoglobulin, in the case of IgG, or changes in plasma volume in the case of all three isotypes, Ga and M. These changes in monoclonal measurements can in fact result in an underestimation of the production of protein by the tumor. Total IgG measurements and SPE, for instance, which are dependent on concentration, cannot account for these changes, and their values will be affected by the relative concentration of, or the relative amount of plasma and hematocrit in these patients. Okay, let's talk about the use of Hevylite at diagnosis. Now, one of the things that we commented on earlier was that not all monoclonal proteins migrate appropriately on an SPEP. And some of the data to back up that statement, other than your personal experience in the laboratory, is this data published by Wang et al. About 6 years ago. They looked at a variety of M proteins and then categorized them by type and where they migrated. The type of M protein is listed down the left-hand side of the table. As you can see, most of the IgG M proteins actually migrated appropriately. Only 6% migrated not in the gamma region. For IgA, however, you can see that in this particular study, 58% of the over 400 patients tested migrated outside the gamma region, primarily in the beta region of an SPEP, where their concentration might be obscured by proteins such as transferrin. You'll also notice down at the bottom of the slide that free kappa and free lambda also have a marked tendency to migrate outside the gamma region. This, combined with their relatively lower concentration compared to the intact immunoglobulin proteins, could easily be the reason why only about 50% of light chain only myeloma patients are detectable by an SPE. On this particular slide, you see an SPEP gel analysis of 14 IgA myeloma patients at presentation. Down the left-hand side of the gel, you see normal human serum with a dense band at albumin, available and visible bands at alpha 1, alpha 2, and beta, and a heterogeneous smear of diverse immunoglobulin in normal serum during the gamma region. The other 14 lanes of the gel are samples taken from known IgA myeloma patients. As you see, with the patient, for instance, on the right-hand side marked by the red arrow, that's a patient where we can imagine that the monoclonal band is in fact migrating more or less within the gamma region. But as you look across the rest of the gel, in the cases where bands are apparent, you can see that many of them migrate outside the gamma region. If you, for instance, look in on the fourth lane from the right, you'll see that there's a very dense band on this IgA myeloma patient, but it's migrating exactly in the beta region where its total concentration would be obscured by the presence of endogenous transferrin. If you now look at the three patients in the middle labeled 1, 2, and 3, I've circled the gamma region on an SPEP where there's really no visible band there to be had. These patients do have an IgA myeloma, but judging from their SPEs, you'd have a difficult time making that call. However, when we plot these patients along with others on the Hevylite assay, we see several interesting things. This is data from a total of 35 IgA patients taken from an MRC trial in the UK, and their IgA kappa is plotted on the x-axis along with their IgA lambda value on the y-axis. The small blue diamonds in the middle of the graph denote the normal range for Hevylite IgA, kappa and lambda, and the two lines that run through on a diagonal denote the upper and lower limits of the A kappa to A lambda ratio. A couple of things show up. First of all, you see that patients 1, 2, and 3 that were not visible on the earlier SPE are now readily distinguishable from normal using their Hevylite concentration. So as you can see, patients 1 and 3 are IgA kappa patients, and patient 2 is an IgA lambda patient. Now, one of the things in the early days of Hevylite that we got from some investigators was, well, for IgA, I'll just monitor their total IgA concentration. After all, it's all the IgA they're making for most of these patients. Well, the normal range for total IgA is represented in the box in the upper right of the graph. And as you can see, looking for patients 1, 2, and 3, if you total their A kappa plus A lambda, the values that are listed underneath the symbol, you can see that for patients 1 and 3, they fit completely within the IgA normal range. Patient number 2 is about 3% above the normal range. So if you were measuring by SPE, all these patients would look normal. If you were measuring by total IgA, they would all be normal or so small above normal that you probably wouldn't be able to see this and distinguish it as being an abnormality. But patient number 3 has an A kappa to A lambda ratio of 15 with an upper limit of normal of 2. Patient number 1 has an A kappa to A lambda ratio of 40, again with an upper limit of 2. Patient number 2 has an A kappa to A lambda ratio of 0.15, where the normal range, lower limit of the normal range, is 0.78. So as you can see, simply using Hevylite, all of these patients were readily distinguishable from normal when they would not have been by using SPE or total immunoglobulin. This is just further data using a larger patient cohort from the French IFM 2005 trial. This retrospective study looked at a total of 94 IgA patients, all of whom were readily distinguishable from normal, either by their concentration of involved immunoglobulin heavy light chain pair or their A kappa to A lambda ratio, or in most cases both. Similar data from the same trial looking at 225 IgG patients again shows that these patients are all distinguishable from normal. In the words of the authors, 100% of the patients were distinguishable from normal using the Hevylite assay. I think if you look closely at the 3 open circles just above the normal range ratio for the IgG lambda patients, and the 2 or 3 circles very close just below the normal range for the IgG kappa patients, you might have trouble distinguishing those from normal, but 97% or more of these patients were readily distinguishable from normal using Hevylite. So let's talk now about monitoring response to therapy. One of the things that we have to deal with when using SPE as a monitoring tool is that if we have successful therapy and a patient is approaching normal, we know that at low concentrations of monoclonal protein, SPEP has a higher CV than it does with higher concentrations. This was demonstrated by Jerry Katzman and coworkers at the Mayo Clinic in an article published about 3 years ago. As you see from this data, below 10 grams per liter, which is approaching normal for IgG but still markedly abnormal for IgA or IgM, you can see that the CVs in terms of serial measurements on these patients now approach 20 to 25%, giving us a wide variance in the results that we might be getting from a patient at any one time. Let's look now at a couple of case studies. In this case, we'll look at serial analysis of a patient with an IgG lambda myeloma being monitored over time during therapy using total IgG in the blue diamonds, monoclonal IgG in the brown circles as measured off densitometry traces from the SPE, and in pink, the pink squares, the heavy light chain ratio. Now, for purposes of this graph, the heavy light chain ratio has been flipped, and what's being plotted is G lambda over G kappa, something that we would normally not do, but it makes it fit on the graph in this case. The SPEP gel that is giving us the monoclonal IgG measurements is represented across the bottom of the slide. What you see here is that beginning at diagnosis, we have a patient with a total IgG concentration slightly outside the normal range, with a monoclonal spike of about 15 to 20 grams per liter, and with a heavy light chain G lambda over G kappa ratio of about 10. As the patient is being treated, all 3 analytes decrease along the same time frame because they measure variants of the same analyte, monoclonal IgG. However, when you reach around day 200, a couple of things have started to happen. One is that the total IgG has gone into the normal range and remains relatively flat, for the next year and a half. About day 350 is the last time we can see a band on SPE and therefore get a monoclonal IgG reading off of the densitometry trace. We then enter a gray area represented on the graph in which both the IFE and the SPEP were read as not abnormal by the pathologist, but we can see following Hevylite that we can actually follow the patient down into just below the upper limit of normal for G lambda over G kappa. The patient then stayed there for the subsequent 2 measurements, and then probably around day 600 we see the beginning of relapse in a slight rise in this ratio. By day 750 or so, we're now seeing a positive Hevylite chain ratio. The SPEP is finally caught up and is able to give us some value. But what happened here is for a period of almost a year, we got no useful information out of total IgG, immunofixation, or SPE because of the limits of detection or the analyte they were looking at. However, during this time, for 3 measurements, we had confidence that the patient was actually within the normal range because we could measure the IgG lambda over IgG kappa ratio. At the 4th point, we start to see what turned into be the beginning of relapse. This is confirmed a little bit more by this data that was published last year by Heinz Ludwig and co-workers. This is a patient with an IgA kappa, and if you look on the left-hand side, in the upper part of the figure, you see the SPEP traces as we go across from left to right, measuring a patient over time. In the middle, you see the gels from the immunofixation, and below that, you see the Hevylite chain results. Now, when you're looking at Hevylite results, we've color-coded this so that red is abnormal and black is normal, just to make it a little easier to follow. When the patient presented, we see a prominent M spike. We see a distinct band that calls the patient IgA kappa on the IFE. And we can see that his A kappa concentration is about 45 grams per liter, and his A lambda concentration is 0.1 grams per liter, giving us a ratio of 420. At the next measurement, following the initiation of treatment, we can see that the patient has started to successfully respond to therapy. There's a smaller spike. There's still an A kappa band on IFE that hasn't really changed because we can't quantitate off IFE. But we see by looking at the Hevylite results that the A kappa concentration has been cut almost in half, down to 25 grams per liter, and that the A lambda concentration is actually doubled in this patient. The result of that is that the ratio has now dropped by 4-fold to 110. The following patient visit, the SPEP was normal, or not abnormal. The immunofixation was negative. But we see that the A kappa and A lambda concentrations yield a normal ratio of 1.36. The concentrations are still abnormal. They're below normal for this patient, probably due to the toxicity of the therapy just wiping out both the involved and the uninvolved plasma cells. The following visit, the SPEP and IFE continue to yield no results. The Hevylite results have returned within the normal range, although they're just barely over the threshold of the lower limit of the normal range. The ratio is solidly within the normal range at 1.4, indicating that although the patient has a lower level of plasma cells overall for IgA, he clearly has maintained a normal ratio. The interesting part comes within the next gel, or the next patient visit, where the SPEP and the IFE continue to yield no data. The IgA kappa concentration for this patient, the original monoclonal protein, is within normal limits. However, you can see that the A lambda concentration has dropped just below the normal range, giving us a ratio of 3, A kappa to A lambda. Remembering that the upper limit of normal is 1.78, we now see that this patient may be in the first stages of relapse, not represented by the presence of its of a large amount of its monoclonal protein, but by the fact that the A kappa tumor is now pushing out the uninvolved A lambda plasma cells, giving us a low a lower level of A lambda and therefore a higher ratio. By the following visit, there's a modest spike on SPE, and the IFE has returned to yielding a result being called A kappa. And you can see that the Hevylite ratio has gotten worse, and both immunoglobulins, the involved and the uninvolved, have gone outside the normal range. However, the important here is that there was a 5.5-month lapse between the time Hevylite began to give us the first indication of relapse and IFE caught up. Now, in the last visit, you can see that the patient has, in fact, started to progress into even worse disease, and the Hevylite ratio has actually gone up to nearly 8,000. I want to turn now to a study that was presented at last year's ASH meeting just this past December. This study, using data and presented by Ola Langren's group at the NCI, looked at a group of 50 patients, 38 multiple myeloma and actually 12 smoldering myeloma patients, who were being treated with a carfilzomib Revlimid low-dose dexamethasone therapy. In this study, 33 of these patients achieved some form of a complete response, 22 reaching the criteria for a stringent complete response, and 8 reaching a near complete response. What Langren and co-workers chose to do was measure the number of CRD cycles that had to be administered to achieve the first CR of any kind. In 15 of these patients, the first CR was achieved within the first 4 cycles of therapy. In 12 of the patients, it was between cycles 5 and 8, and in 6 of the patients, more than 8 cycles of therapy were required. 17 patients failed to achieve a CR of any kind. When looking at the relationship between normalization of Freelite and Hevylite chain ratios and the time that it took to get the patient to CR, Butani noticed a rather interesting association. As you see at the bottom of the graph, of the patients that entered CR within the first 4 cycles of therapy, 13 out of 15 of those patients normalized their Freelite chain ratio after a single cycle of therapy. If a patient's Freelite chain ratio normalized, represented in red, after more than 1 cycle of therapy, it in general took those patients longer to achieve CR. Similar data was generated using the Hevylite antibody. Now, in this case, we needed to look at after 2 cycles of therapy, and just remember that Hevylite measures an analyte with a much longer half-life of days to weeks than the short half-life of hours of the Freelite reagents. What you see here again represented in blue are those patients who achieved a normalization of their Hevylite chain ratio after 2 cycles of therapy, and you can see that all of those patients normalized or achieved CR within the first 8 cycles of therapy, most of them within the first 4 cycles of therapy. Patients represented in red who did not normalize their Hevylite ratio until after 2 cycles of therapy took longer in general to achieve a CR. Now, one of the questions that comes up is, well, we have a lot of tools for measuring in the laboratory. Do we really need to use all of them? I mean, after all, laboratory testing is expensive and healthcare is under constant scrutiny for trying to find ways to lower costs. Well, in the case of multiple myeloma, we know that the drugs that we use cause the tumor to evolve. But first of all, and I'm sorry, I got a little ahead of myself there, I want to come back to the point I made about does Freelite replace Hevylite? Or does Hevylite replace Freelite? And the answer is no, they measure different things. Now, as I told you, Freelite measures free kappa and free lambda light chains not associated in intact immunoglobulin, whereas Hevylite measures intact heavy chain light chain pairs. This data that came out of the French IFM study looking at 164 patients with an IgG kappa myeloma plots their Hevylite IgG kappa values across the x-axis and their free light chain kappa values across the y. And as you can see, as this group of patients, there's really no association between the amount of heavy light chain being produced, the amount of intact M protein being produced, and the amount of free kappa and free lambda. Both are biomarkers being made by the tumor, but the levels do not relate to each other. So this is the data I was alluding to. This and the next slide are the data I was alluding to when I talk about clonal evolution. This is data published by Keats et al. In the group from Mayo Scottsdale about a year and a half ago now, looking at how patients responded to therapy. What's represented on this slide is the result of a large analysis done on each individual patient, looking at their tumor subpopulations. These subpopulations were defined using flow cytometry, fluorescence in situ hybridization, PCR, and a variety of other tools. Well, what you see out of this, represented in colors as a pie chart, is, for instance, if you begin at the 9 o'clock position on this graph, you can see that about 3/4 of the tumor load for this patient was represented by the clone designated 1.1. However, there were certainly at least 3 other noted and distinguishable subclones within this patient that had a different profile when using these multiple tools. As the patient was put on lenalidomide and dexamethasone, we can see that in the first remission, that the level of the clone 1.1 represented in red had dropped somewhat, and an expansion of clone 1.2 represented in orange had tripled. The patient was maintained on a Lendex regimen, but went into relapse, at which time there was a marked increase in the percentage of clone 2.1 represented in green. The patient was then switched to a carfilzomib therapy, and you can see that clone 2.1 appears to respond to carfilzomib, but clone 1.2 now becomes a much larger percentage of the population. This trend continues until the patient finally dissolved into a plasma cell leukemia, where the bulk of the tumor load was now what had been a relatively minor subclone of these for this patient. What this is pointing out is that as we continue to treat tumors with different regimens, that different populations may be sensitive or insensitive to the tumor, to the chemotherapy that's being used, and that any single tool may give us a false sense when it comes to how the patient is responding. This was also looked at using somewhat more conventional tools or more traditional tools by Zamarin and co-workers at Memorial Sloan Kettering. This looks at a group of patients who are all treated with the same kind of therapy, and as you can see from this graph, about half of the patients of the 126 cohort achieved a CR during therapy, and about half did not. Looking at the type of M protein present at diagnosis using the Freelite assay and SPE, we can see that the two populations were relatively the same at diagnosis. That is, about 20% of the patients presented as light chain only myeloma patients measurable by Freelite. About 60 to 70% of the patients represented a mix of M proteins, such as represented in red here. This has mean they were positive for both the SPE and Freelite assays because they were making both intact immunoglobulin and free light chains. And represented in green, about 10% of the patients made intact immunoglobulin only and had no detectable abnormalities using the Freelite test. So you can see going in that we already have a diversity of patients who are measurable either by SPE or Freelite alone, or both. However, when these patients relapsed, which they all did, unfortunately, we can see that the pattern starts to change. One is, so the patients who started with Freelite only all relapsed as light chain only patients, measurable by Freelite. The patients who went in with a mix of M protein production came out with an even bigger mix. As you see, for the patients who achieved CR on the left, almost half the patients in this group downregulated their free light chain production and were measurable by SPE, making intact immunoglobulin. However, as you can see, about 11% of these patients converted to a phenotype where they were only making free light chains, represented in blue. For the patients who had achieved something less than a CR upon initial therapy, you can see that in fact about 25% of those patients stopped making excess light chains as measurable by Freelite, but 38% of the patients now made light chain only. The patients who started with an intact immunoglobulin only production came out for the most part continuing to make intact immunoglobulin only, but as you can see, 17% in both groups now started making excess free light chains as well. So there's something to be said in here. There's something that's being told to us about plasma cell biology and responses to chemotherapy. And we're not quite sure what this means yet. But what's also clear from this is that if we're only using a single tool, such as either SPE or Freelite or Immunofixation, we're going to miss patients, either at diagnosis or at relapse or both. Now I want to add right now that Hani Hassoun, who was the principal investigator on this study at Sloan Kettering, is currently looking at the same patient cohort using the HeavyLight assay as a replacement for SPE in this particular group of patients, especially in the relapsed group, and we're very excited about the prospects that might come out of that study. Okay, so then the question becomes, well, you've got these tests now in addition to SPE and IFE, we've got HeavyLight and Freelite. How do I put all this together? Now, this is not an algorithm that has been published in a guideline at this point. It is, however, a suggestion of a potential algorithm for testing myeloma patients. If you have a patient with a suspected monoclonal gammopathy, we know that data published by Jerry Katzman and coworkers at the Mayo Clinic has indicated that the combination of SPE and free light chain will detect all monoclonal gammopathy patients, all symptomatic monoclonal gammopathy patients, at diagnosis. That if the patient with a suspected monoclonal gammopathy due to the symptoms has an unremarkable SPEP and a normal free light chain ratio, that patient does not have a monoclonal protein disease and can be evaluated for other causes. If, however, either one of those tests results in an abnormality, then we need to reflex to an IFE in order to identify the isotype. This is primarily for efficiency, as it will let us run in one test and determine what the isotype is of the monoclonal protein being produced. At that point, we can now use HeavyLight to establish a baseline for the appropriate heavy light chain pair. So we don't need to run all three. We can simply run A kappa and A lambda for an IFE that says the patient is A kappa or A lambda, or IgG for an IgG patient, etc. That lets us now monitor disease in response to therapy using the combination of SPE, HeavyLight, and Freelite. So I hope what I've been able to convey today is that HeavyLight recognizes unique conformational epitopes which can distinguish between heavy chain, light chain pairs. That this enables the clinician in the laboratory to quantitate involved and uninvolved intact immunoglobulin in myeloma and related plasma cell dyscrasias. That the heavy light chain ratio can quantify disease by measuring involved against uninvolved immunoglobulin in ways in which total immunoglobulin isotype measurements cannot. And that the heavy light ratio can be measured at levels below the detectable range for either SPE or IFE. Again, heavy light, like free light, can read all the way into the normal range and therefore gives us sensitive detection for patients who are either entering remission, have minimal residual disease, or are in the early stages of relapse. Data that I didn't have time to show you today, but using HeavyLight, we've indicated that like the free light ratio, combining HeavyLight with other staging criteria can enhance risk stratification in newly diagnosed patients. And with that, I would just remind you that we have some time to take a few questions. There is the Q&A button in the lower left section of your screen, and I'm now going to turn it back over to our moderator so that we can start to address any questions that might have come in.
Thank you, Richard, for that very interesting webinar. Audience, the session is now open for questions. You may ask a question by typing the question into the question and answer box located in the lower left hand of your screen. The first question I have is, does HeavyLight and Freelite mean we don't need to do MRD testing, e.g. via flow cytometry?
Well, that's actually an interesting question, and I don't think that's going to be the way that this shakes out. We won't know until groups such as the International Myeloma Working Group have put together guidelines. But I think what it's more likely to be is that HeavyLight and Freelite may give us sort of a gateway into which tests patients should be evaluated for MRD. I think it's easy to see that if a patient, for instance, since we know that Freelite and HeavyLight are going to be cheaper ways to test for this patient, for whether or not they have minimal residual disease, that if we use Freelite and HeavyLight as the gateway by running those tests ahead of time, if both of those tests are normal, that's a candidate for looking at flow cytometry, which is very sensitive, obviously. But if those tests, either one of those tests is abnormal, I think it's fairly safe to say at that point that that patient would not be a candidate for flow cytometry evaluation of MRD. They're just not there yet.
The next question I'm sorry, I'm just opening this up. Does this test provide a measurement of M spike, i.e. myeloma protein?
Yes, it absolutely does. We can measure what the HeavyLight result you would get. Let's say a patient is an IgG kappa patient. There's three pieces of data that will come out of there. If you use HeavyLight IgG reagents on a patient with a G kappa myeloma, you'll get a measurement of the M spike in the sense that you'll get an IgG kappa concentration from that patient, and that you can compare against the normal range. You'll also get a measure of the uninvolved IgG lambda concentration, and we know, for instance, that in patients with G kappa myeloma, that their G lambda plasma cells are suppressed in the bone marrow by the tumor. The third piece of information you'll get is the ratio between G kappa and G lambda. So you'll be able to compare not only the size of the spike using G kappa, but the uninvolved G lambda concentration and the overall clonality of the patient by taking the ratio of G kappa to G lambda.
Thank you. You displayed reference intervals with a 2D graph. Can the reference intervals be adequately documented with a single numerical range for each analyte?
Well, I think the answer, comes not only from the fact that we've done a fair amount of testing in a variety of normal populations, and they all fall within that normal range. So, understand that the graph that we displayed that had a reference interval not only had the ratio between any particular analytes, kappa and lambda, but it also had the concentration of each. So in reality, there's three normal ranges if I display it on the graph like that, because for each data point, there's a concentration of, for instance, A kappa and A lambda. These were all the normal ranges that we put into the FDA submission. They've been looked at by several different investigators. I should comment at this point that HeavyLight has actually been FDA approved in Europe for over a year and has been widely used, especially by the French group. And so I think we're pretty confident about the normal ranges that we have. Clearly the FDA bought into it, which is always important.
Thank you. The next question is from Zhang Xiao, a clinical chemistry fellow from Houston Methodist Hospital. Two questions. The first question, could this HeavyLight assay diagnose heavy chain disease? The second one is he wants to know if he can have your slides, and this will be posted online for a while, so I can answer that one.
So it's actually an interesting question, and I would say right now the answer is as best we understand, it probably would not work very well in heavy chain disease. And that is because the reagent recognizes combination epitopes of heavy and light chains. This is not a reagent that recognizes IgG. It recognizes IgG kappa specifically. So it recognizes conformational epitopes formed between the pairing of G kappa and G lambda.
Thank you. The next question, are there any efforts at this time to incorporate the HeavyLight assay into the international guidelines for myeloma response?
So that's a great question, and the answer is yeah, we're absolutely working on it. Binding Site has a history of working very closely with the International Myeloma Working Group. Many of the members of this group are enthusiastically looking at HeavyLight in clinical studies right now, many of which we hope will be published this year. And I know that they are now looking at whether or not this should be incorporated into a revision of the guidelines for diagnosing and monitoring plasma cell dyscrasias.
Our next question, could HeavyLight replace IFE?
I'm going to say the answer to that really is yes and no. And the way I would suggest that it be used is you probably aren't going to want to run HeavyLight right up front when you know simply from, say, let's say you ran SBE and Freelite to diagnose whether or not you had a patient with a monoclonal gammopathy. If you get a spike on an SPEP or an abnormal Freelite ratio or concentration, you still don't know what the isotype of that intact pair, whether or not it's an intact Ig pair, and whether or not, and what the isotype of that pair might be. So what we'd suggest at that point is that you run a single immunofixation at diagnosis in order to determine the isotype of the monoclonal protein that you're dealing with. Now, while I'd love to tell you, and my marketing department will be more than happy to say that, well, if you ran all 6, that would be great because you'd get the same information, the reality is that's not particularly efficient. So if you run a single IFE at diagnosis to determine the isotype of the patient, you'll be able to tell, for instance, if it's a light chain only myeloma, then you don't want to run HeavyLight, but you certainly want to run Freelite. But if the gel then comes back and says, well, it's a G kappa myeloma, then you know that if you run G kappa and G lambda HeavyLight, you'll get the information you need. What we know right now is that it doesn't appear that there's a lot of data to be had by looking at the other isotypes. So for instance, looking at A and M in an IgG patient by HeavyLight is not particularly useful, but using the HeavyLight G kappa G lambda testing provides very valuable information.
Thank you, Richard. I just have a little information. The slides are available for downloading on your screen by clicking on Content and Files. The PDF is there. We still have a couple more questions that I do want to address as well. The next question, as per your suggested myeloma algorithm, does this mean that urine is no longer necessary, or are there selected cases where urine would be necessary?
So this is sort of an ongoing discussion. The reality of the situation is it's been published for quite some time now that what we measure in urine can be measured in serum. So for instance, when we talk about Freelite, we know that patients who have, that if you have, for instance, Freelite chains in your urine and therefore a resulting positive test for Bence-Jones proteinuria, that those light chains started off in your serum, that they were filtered out by the kidneys and ended up in your bladder as a result of their initial production and presence in the serum. There's been data published for quite some time now where Mayo and several other groups have looked and said that any patient that's Bence-Jones positive, we can detect using serum Freelite. So from that standpoint, they don't really necessarily need urine at diagnosis. I do want to point out, however, that the international guidelines say for multiple myeloma that you don't need urine at diagnosis in order to monitor or in order to declare the patient as having multiple myeloma, but urine proteinuria is still being used as a measuring metric for patients with AL or primary amyloidosis. So the answer is it's probably not necessary because you're going to detect anything in serum that will eventually end up in the urine, but the reality is current guidelines indicate for amyloidosis that it's probably true. I didn't show you any data today on HeavyLight and amyloidosis. There's actually some very exciting data that's being put together looking at the utility of HeavyLight in AL amyloidosis, and hopefully in some future broadcast we might be able to talk about that.
Just a couple more questions. The next question, what is the clinical utility for total light chain measurement?
Short answer, none that I know of. I've asked multiple clinicians, some of whom still use total light chains, why they're using it and what does it tell them. And the primary answer I get back is tradition. Those sites that are using total light chains, there's no indication, there's certainly no guideline published, there's no utility that I've ever been able to detect that total light chains give you any information that is not there. In fact, if you look at the relative concentration, so if you're measuring, for instance, kappa light chains, or if you're measuring total kappa in a patient with either an IgG kappa or a kappa light chain only myeloma, you're not only measuring the kappa that's associated with IgG, you're also measuring the kappa that's associated with IgM, with IgA, with IgD, with IgE, and the free kappa. And there's just such a cloud in there that it's really hard to tell what you're getting. If you look at the concentration of free kappa versus total, it's literally 500. Free kappa is present about 500 to 1,000-fold less in concentration than total kappa or total lambda. So, and there's no guidelines out there anywhere or any data that I'm really aware of that say total kappa or total lambda provide any meaningful measurements in any sort of plasma cell dyscrasia or monoclonal protein measuring.
Thank you. The last question is a twofold question. It is, what is the reimbursement status of HeavyLight, and where is HeavyLight available to order today?
Okay, so HeavyLight is reimbursable under Medicare and private insurance. It's actually going to be billed out, I think, for those of you who are used to it, as a similar code, the same code that's used for Freelite in terms of, I don't remember the exact CPT code, but it's nephelometry by other instruments. It's the same CPT code that's used for Freelite. In terms of where it's currently available, Mayo Medical Laboratories are running HeavyLight now, so it will be available as a send-out through any local hospital who send out to Mayo. And LabCorp, we believe, will be live for running HeavyLight within 30 days, if not less. I've talked to the folks at LabCorp personally, and they're just finishing up dotting the I's and crossing the T's on their protocols and should be beginning to offer the test, as I said, within 30 days. So,
Thank you. Audience, thank you for all your questions. Thank you, Dr. O'Hara, again, for this very informative and educational webinar. If you have any questions regarding anything you learned on this webcast or would like to request any additional information, please feel free to visit our booth here at Clinical Diagnostics and Research Bioconference Live virtual event. Just click on Binding Site under the Exhibit Hall directory. You can also contact us by email at info@thebindingsite.com or find more information on our website at www.thebindingsite.com. Thank you.